1 in 230 cats are diagnosed with feline diabetes, but many more likely remain undiagnosed. Feline diabetes is incredibly hard to identify. By the time owners have recognized typical symptoms, cats will have to undergo a lot of stressful tests, including urinalyses and many blood tests, but even then, it may be too late. For these reasons, it is incredibly crucial for there to be an early diagnosis of feline diabetes, as it can be most effectively treated in its earlier stages. We are RHS-Calgary, a group of extremely passionate high school students, and we created GlucoLitter. GlucoLitter is designed to make it easy and efficient for cat owners to monitor their pet's health. The way it works is simple. When a cat urinates in a litter box, its owner can scoop up some of the litter and test it using our product. Through fundamental synthetic biology principles such as the use of enzymes, biochemical reactions, and a simple colorimetric assay, a color change will be produced to indicate the presence of abnormal amounts of glucose. If there isn't any change in color in the litter, there is nothing for the owner to worry about. However, if it turns purple, it is indicative that the cat potentially has abnormal amounts of glucose in their urine. As stated, this is crucial in the early diagnosis and prevention of feline diabetes, and it could make it efficient for owners to notice if their cat should be taken to the vet. Let's talk a little bit more about the synthetic biology behind GlucoLitter. Diabetic cats are unable to filter excess sugars out of their systems, causing it to pass through their urine. Due to this, the easiest way to determine whether or not a cat is diabetic is simply by analyzing the level of glucose in a urinary sample. With this in mind, we came up with our experimental design. The modeling of our experimental design begins with a reaction between beta-D-glucose and oxygen, which will then produce D-glucono-1,5-lactone and hydrogen peroxide in the presence of an enzyme, glucose oxidase. The hydrogen peroxide will then form another biochemical reaction with a colorimetric assay kit containing the dye xylenol orange in order to produce a purple pigment. Essentially, abnormally heightened levels of glucose will trigger a reaction catalyzed by glucose oxidase, producing hydrogen peroxide and hence expressing a color change. The distinct color change from yellow to purple can then indicate to a cat owner that the levels of glucose in their pet's urine is of concern. One of the key aspects of our project, which we are currently testing through prototyping, is the concentration of glucose required to be present in a cat's urine to deem it healthy versus diabetic. Our team is grateful to have been mentored by a vet technician, Miss Emily Chorwood. She provided us with a few important pieces of information that greatly aided us in our calculations and usage of biological components. Firstly, when the blood glucose level exceeds 10 millimoles per liter, it is excreted in the urine, so anything above this amount is considered concerning. Secondly, she provided us with a gradient of glucose concentrations that veterinarians would generally associate with the different stages of diabetes in felines. These numbers were crucial in all of our calculations, specifically when determining how much enzyme and working reagent would be needed in order to express accurate color-changing results in a glucose solution that simulates that of the urine of a diabetic cat. In the many weeks preceding the cGEM competition, our wet lab team has been diligently working to create a sound proof of concept which demonstrates that our product will yield successful results. Through vigorous experimentation, we have determined that our target reaction would in fact produce a color change in solutions containing high levels of glucose, which was then further confirmed by various controls that we included in our experimental design. Currently, we are working on determining concentration gradients for GOX in order to make our reaction more effective and efficient. However, we stumbled across some challenges throughout our many experiments, one of which was that our controls changed color when in fact they should not have. For this reason, we came up with a well-rounded hypothesis stating that hydrogen peroxide is volatile in nature and trace amounts of its vapors reacted with other substances in their aqueous form. As a result of the controls being exposed to H2O2, they also in turn changed color. Since then, we have remodeled our experimental design and have been producing accurate results. Although we are still in the prototyping stages of our experiment, we have carefully constructed a detailed layout of our experimental design so we can further our research in the forthcoming months. We plan to construct a plasmid using the genome sequence of the GOX enzyme. We acquired a his-tagged sequence of GOX from an Israeli iGEM team that previously conducted independent research on it. The plasmid we construct will then be transformed into BL21 E. coli bacterial cells. This particular strain of E. coli is specifically engineered to efficiently express high levels of protein. After the transformation of BL21 with the constructed plasmid, the cells will be induced, and through the processes of transcription and translation, a GOX protein will be synthesized. Finally, we will conduct a protein purification protocol to isolate the GOX enzyme for our designed assay. Once we have genetically engineered the protein, we will develop our own colorimetric assay kit.
While the wet lab has conducted experiments that are vital for a product to thrive, none of it would have been possible without the diligence of everyone on our team and the guidance of our mentors. Our team, subdivided into wet lab, dry lab, and human practices, all work together to bring our ideas to fruition. One of our goals as a team was to foster and maintain an interdisciplinary environment. We have done this through consistently ensuring that we hold meetings between all of our sub-teams. This way, everyone is aware of the progress that we are making as a collective. A large part of our work also involved collaboration with individuals outside of our school's team. Particularly, we have fostered a relationship with the University of Calgary iGEM team, with whom we have received mentorship and feedback from. We have also worked with countless people in the industry, including researchers, veterinarians, and cat owners themselves. First, as we have mentioned previously, we are very grateful to have been in contact with the vet technician, Emily Chorwood, who provided information that was key for our experimental design. We have also worked alongside Dr. Laura Kiefer-Wilkes, who provided us with guidance and held multiple workshops so that we could gain a fundamental understanding of the main concepts in synthetic biology.
Finally, a large contributing factor to the overall design of our project comes from owners of diabetic cats. Specifically, we have spoken to Mr. John Whitbread about his personal experiences. He provided us with mentorship on owning 2 diabetic cats and the maintenance it involved. He particularly shaped the way that GlucoLitter was formed by offering insight on what he believed to be the most effective, efficient, and humane methods of testing. From the get-go, he agreed that GlucoLitter would make it much simpler for cat owners to notice the symptoms of diabetes. Initially, we were focused on creating a litter additive directly incorporated into the litter. Mr. Whitbread expressed, however, that cat owners may be concerned about the health risks associated with their cat being directly exposed to our product. With this, we switched to a spray-on version, meaning our product is stored in liquid form and is sprayed onto sections of the litter when removed from the box. Not only would this create an even safer environment for the cat, but it is also much more consumer-friendly.
From this, we also conducted a market analysis. Some products that are currently existing are Pretty Litter and Ultra Monthly Monitor. Pretty Litter is a litter additive that is useful for looking at common health issues in cats, but it does not take into consideration the testing for diabetes. Ultra Monthly Monitor looks specifically at urinary tract disease in the form of a litter additive. So, it can be said that there is a good marketability opportunity for our product. GlucoLitter is unique in that it would be able to be in liquid form. and would test specifically for feline diabetes.
Even veterinarians, such as Miss Emily Chorwood, have agreed that GlucoLitter has the potential to change many lives. Along with interviewing people like Mr. Whitbread and Dr. Laura Kay, we have also been focused on reaching out to the general community. As a team, our outreach has been mainly focused within our school. One of our guiding focuses as a team is to advocate and bring awareness to feline diabetes as a whole, as well as to foster a passion for biology in our peers. Previously, we hosted a week-long synthetic biology camp that aimed to introduce younger students to the splendor of biology. The camp was geared towards creating a foundation for younger students to build their interest in SynBio through opportunities such as iGEM. We have then further involved younger students in iGEM by giving them the opportunity to interact with the team, help, and observe how iGEM works. Many cats are suffering each day, and it is our desire to make it possible for cat owners to keep their pets safe, happy, and healthy. By making it possible for there to be early remission and diagnosis of feline diabetes, we can help save countless lives. With GlucoLitter, we can provide cat owners with the tools that they need to monitor their pet's health at home in a safe and effective manner. This is GlucoLitter, saving lives one colorful litter box at a time.